Hello, my name is Saurabh Garg. I'm a scientist in Dr. Brian Czerniecki's group at Breast Oncology. Today I'm presenting at Moffitt Symposium I'm presenting a poster, and the title of my poster is a correlation of oncodriver expression with tumor infiltrating lymphocytes in triple-negative breast cancer. So our lab mainly specializes in using dendritic cells as a therapeutic agent, and we use Moffitt cell therapy facility to produce these DCs, which we isolate from monocytes and convert them into type 1 polarized mature dendritic cells. In this process, we also introduce MSC class 2 peptides, which are oncodriver specific for a specific tumor. So in this particular project, we want to focus on patients who are having triple-negative breast cancer, and we want to focus on TNBCs because of the reason it is one of the most prevalent form of breast cancer and also represent a very aggressive form of breast cancer with poor prognosis and very limited treatment options. So in this study, what we did, it is a retrospective study of 90 patients. We use a tissue microRNA analysis to analyze the expression of 3 different oncodrivers, MET, EGFR, and HER3, and correlated their expression with the immune infiltration in form of CD4, CD8s, and other immune cell population. What we saw in our findings that MET and EGFR expression was high in tumor tissue in comparison to benign tissue, and EGFR MET expression correlated between themselves, but HER3 expression was mutually exclusive. We did see HER3 expression correlated with chemotherapy status, where patients who received chemotherapy showed more HER3 expression after chemotherapy, suggesting a possible involvement of HER3 in resistance mechanism. The main finding of this study was that all 3 oncodrivers, HER3, MET, and EGFR, expression was correlated with some form of immune infiltration. For HER3, we have more CD4s where HER3 expression was high. For MET, we have more CD8s where MET expression was high. And for both MET and EGFR, we saw more activated lymphocytes in TNBC samples. So it basically gave us a good indication that these oncodrivers have some immunogenic regions which we can harness for our DC-based immunotherapies. And so the future of this study would be to identify those immunogenic regions from these oncodrivers and use them to identify those peptides, screen those peptides, and use them in future clinical studies at breast clinic.